Population Profile, the Vibrio vectorius, by Kenesha McNeil. Targeted population. The scientific name is the Vibrio vectorius. The common name is the Vibrio. An article that I researched on this topic is the Vibrio vectorius, a potential living antibiotic to control bacterial pathogens, by Cavell, published in 2021. The article is discussing the potential of the Vibrio vectorius as a biocontrol agent against bacterial pathogens, highlighting the unique bacterial lifestyle and applications in combat antibiotic-resistant bacteria. Geographic boundaries. The Vibrio vectorius is a uniqueness found in diverse environments, including soil, fresh water, marine ecosystems, sewage, and even within intestines of animals. For this study, we focused on the freshwater ecosystem within Albemarle Pamlico systems in North Carolina, United States. This region composes various freshwater, brackish water bodies, providing a suitable habitat for the Vibrio vectorius and its negative gram bacterial prey. Data collection methods. Demographic data. Sense data. Population size. Quantity that Vibrio vectorius populates using a qPCR targeting specific genetic markings. Agent structure and generation time. Laboratory cultures can be used to determine generation times under controlled conditions, typically ranging within 3 to 4 hours. Gender descriptions. As a bacteria reproduces asexual, gender description is not applicable. Research data. Life pattern. The Vibrio vectorius exhibits a biophasic life cycle, alternating between a free-living attack phase and a growth phase within gram-negative by its shootup flagellum. The prey is composed of range prior gram-negative bacteria, agent understanding the equal roles. Laboratory experiments can determine optimal temperatures between 28 and 30. Land use data. Utilizing satellite images to assess land using patterns surrounding freshwater bodies in the Albemarle Pamlico systems. Examine correct correlations between land using and their Vibrio population. Resources, consults, and data. Energy source. The Vibrio occupies the role of a secondary consumer, preying on primary consumers, gram-negative bacteria. Waste generations. By lyzing prey cells, it releases organic matter back into the environment, potentially influencing nutrient cycle. Assessing the prey throughout the co-culture, experiments within various gram-negative bacteria strains. Environmental monitoring. Unique managers such as a pH scale, dissolving oxygen temperature and nutrients concentrations. Soil types, analyzing samples for its texture, organic matter content, and microbial divergence. Collecting materials data by understanding the influence on the bacterial population. Pollen levels. Monitor for contamination that may affect the microbial community, such as heavy metals and parasites. Experimental methods. Objective. Determining the beneficial effects of the Vibrio vectorius on local gram-negative bacteria isolated from the Albemarle Pamlico systems. First, you collect a sample from various freshwater sites. Isolate the prey. You collect the gram-negative bacteria from a pond using a selective media. Co-culture experiment. Induce the bacteria into co-culture and contaminate it and isolate it with a prey bacteria such as E. coli. Monitor using density measurements and microscopes over time. 4. Data analyze. Assess the predator range, and environmental factors influence predatory effectiveness. Experimental methods. Assess the results. Using a standard microscope lab in a lab equipment. Also using an autoclaver. Also using a PCR. Other additional requirements. Specify media for co-culture, bacteria victorious, and the prey bacteria. Use a qPCR facility and quantifications. Thank you.